In a dark laboratory, a single strand of light twists upward. Two ribbons wind around each other like a spiral staircase built for something impossibly small. This is DNA. One molecule, thinner than a strand of spider silk, yet stretched out inside a single one of your cells, it would measure roughly 6 feet long. And you have trillions of cells. Packed together inside each microscopic nucleus, the entire instruction book of you fits into a space smaller than a grain of dust. But the scale is not the strangest part. The structure is. Those rungs of the twisted ladder are not random. They are a code. 4 chemical letters, repeated in patterns, spelling out the blueprint for everything: your fingernails, your heartbeat, the shape of your eyes. A molecule that writes. But who wrote the code? And why does it feel less like chemistry and more like an ancient message waiting to be read? To answer who wrote the code, we first had to see the shape. And the person who saw it most clearly was not searching for a message. She was searching for a pattern. In the early 1950s, in a cold London laboratory, a 31-year-old crystallographer named Rosalind Franklin was doing something that looked more like detective work than biology. She would take a sample of DNA, so thin that thousands of strands side by side would fit inside a single human hair, and stretch it into a thread. Then she aimed an X-ray beam at it. The X-rays bounced off the atoms inside and hit a photographic plate, leaving behind a pattern of dark arcs and crosses. To an untrained eye, it looked like a smudged fingerprint. To Franklin, it was a mathematical map of a molecule. She worked relentlessly. One single sample stayed in front of the X-ray machine for 100 straight hours while she adjusted the angles. Her hands were stained with chemicals. Her lab coat was permanently blotched. And in that lab, she was not allowed to eat lunch in the common room where the men gathered. She was a woman in a world that treated her as support staff, even though she was the one doing the hardest work. The breakthrough came when she captured an image, now known as Photo 51. It showed a clear black cross of reflections, an unmistakable signature of a spiral. Franklin understood exactly what it meant. She wrote about it in her lab notebook. The molecule was a helix, 2 strands winding together, but she never got to tell the world. In February 1953, one of her colleagues showed Photo 51 to 2 men at a nearby lab, Francis Crick and James Watson, without her knowledge or consent. They saw what Franklin had worked months to decipher, and they built their famous model of the double helix on her data. When their paper was published, her name appeared nowhere in it. Hers was published later, framed as a minor detail. She died in 1958, at age 37, from cancer, likely caused by the radiation she had used to photograph DNA. She never knew that her image would become one of the most important in biology. But this is not just a story of injustice, because solving the shape only raised a deeper problem. A spiral is just a shape. A code is different. A code carries a message. And now that humanity could see the twisted ladder, we had to ask what it was actually doing. What does this shape do? What is it carrying? And why does it look so much like something we designed? The staircase Rosalind Franklin photographed is not a ladder of identical rungs. The rails are sugar and phosphate, repeating like a bored drummer's beat, but the rungs are not all the same. Each one is one of 4 chemical letters, abbreviated A, T, C, G, and together, these letters create something that does not exist elsewhere in chemistry: meaning. Think of alphabet magnets. Separately, they are just plastic. Arrange CAT and they mean a furry animal. Rearrange ACT and they mean to do something. The plastic never changes. Only the sequence changes. DNA works exactly this way, except the letters are molecules, and the reader is not a human eye. The code is read in groups of 3. Some groups say start here, some say stop. The rest are instructions for linking amino acids into chains that fold into the machinery of your cells. A molecule that builds molecules by reading a text. This is where the comparison to a computer stops being a metaphor and becomes almost literal. A computer processor uses 2 symbols, 1 and 0, to run every program ever written. DNA uses 4 symbols, but the principle is identical. Symbol, not chemical force, carries the information. A chemical bond will always pull the same way. A symbol only means something because a reader has agreed to interpret it. In a computer, we are that reader. For DNA, no one has found the writer. Consider the storage density. A single gram of DNA holds 215 petabytes of data. 215 million gigabytes in a dot of dust. Every film, every song, every page ever typed could fit in a few grams. No engineered hard drive comes close, but a hard drive is not useful without a processor. A program is not a program until something executes it. Inside your cells, tiny molecular machines unzip the DNA, read its letters, and build proteins according to the instructions. Those machines are themselves made of proteins, built from DNA. The code contains the recipe for its own reader. So here is the unanswered question. If DNA is software, and the hardware is also written in DNA, then what ran the very first instruction before any reader existed? That question, what ran the first instruction before any reader existed, was the same problem a man named Claude Shannon was attacking in 1948, but from a strange direction. He was an engineer at Bell Laboratories who rode a unicycle down the hallways while juggling 3 red balls. His official job was pushing messages through telephone wires. But in doing so, he invented a way to measure something nobody thought could be measured: information itself. Not meaning, just the raw quantity of surprise in a sequence of symbols. Shannon's trick was simple. Imagine a coin, 2 sides 50-50. You need exactly one yes or no question to know the result. That is 1 bit. Now imagine DNA, 4 letters, equally likely. You need 2 yes or no questions to identify any single letter. Is it A or T? Is it G or C? 2 bits. Shannon called this measure entropy, borrowing the word from thermodynamics. Higher entropy means more surprise, more unpredictability, more information per symbol. So in theory, each letter of DNA carries 2 bits of information, but real genomes are not random. Count the letters inside a human cell and you find patterns. Some letters appear more often than others. Some stretches repeat themselves. Each letter gives a clue about the letter that follows. The surprise shrinks. The actual entropy of living DNA drops to roughly 1.5 to 1.9 bits per letter. Still vastly more information than any human-engineered code, but lower than the ideal. A message carrying the fingerprint of its own history. Here is where Shannon's ghost enters the helix. His mathematics showed something uncomfortable. Noise scrambles, randomness erases. Uninformed, undirected processes always push a message toward maximum entropy, toward pure gibberish. They never generate new, useful, precise instructions. Yet inside every living cell sits a code that is the exact opposite of noise. It has checksums, repair mechanisms, and a grammar. It stores more data per gram than any hard drive ever built. Shannon gave biology a measuring stick for this wonder, and the numbers are staggering. This quietly transforms the deepest question in biology. It is no longer just, how did these molecules come together? It becomes, how did this much information arise? Because information science says untargeted forces do not write messages. They erase them. And that leaves us facing the origin of the code, with no theory of the code's author. So we are left with the question Shannon himself would have loved. Where did the signal come from? If the code itself is a mystery, then consider what the code builds. Inside the membrane of a common bacterium sits a rotary motor, not a metaphor, a real spinning axle, driven by a flow of protons, attached to a propeller that whips the cell forward through water. This motor reaches speeds of 100,000 revolutions per minute. It can stop, reverse direction, and spin back up in a fraction of a turn. It has a rotor, a stator, a drive shaft, a bushing, and a propeller. It is one of the most efficient motors ever measured. In 1996, a biochemist named Michael Behe was staring at this machine when he decided to ask a simple question. Behe was not a philosopher looking for arguments. He was a professor at Lehigh University who had spent decades breaking open cells and cataloging their parts. What he saw in the flagellum reminded him less of a random assembly and more of a designed tool. So he gave it a name, irreducible complexity. His definition is stark. A system is irreducibly complex if it stops working entirely when you remove any single part. A mousetrap is a good example. Take away the spring, and you do not have a slightly worse mousetrap. You have a pile of wood and metal that catches nothing. The flagellum fits that definition. It is built from more than 40 different proteins, each one folded into a specific shape and locked into a specific position. Remove the stator, and there is no power. Remove the bushing, and the axle slips off-center. Remove any one of the proteins that makes up the rotor, and the whole assembly falls apart. No single part works alone. No subset of parts works as a motor. Every component only makes sense when every other component is already there. And that creates a genuine problem for gradual change. Evolution works one small step at a time, and every step must survive long enough to reproduce. But a flagellum missing a bushing does not spin. A flagellum missing a rotor does not spin. A flagellum missing the proton channel does not spin. There is no half motor that turns slowly and gets faster with each new part. The parts are not improvements to a working machine. They are a team that only functions when the whole roster is present. So the question hangs in the air, unresolved. The DNA code holds the blueprint for this motor. The motor executes precise instructions. But if the motor cannot be built piece by piece, and the code cannot be written by random noise, then where did the first working version come from? No one has answered that. The motor still spins. The code still reads. And we are still standing at the edge of the machinery, wondering who tightened the first bolt. Biologists think they found the hand that tightened the first bolt. It was not a hand, it was a needle. Some bacteria build a structure called the type 3 secretion system. Imagine a microscopic syringe, a hollow tube that pokes through the cell wall and injects proteins into other cells. It is a weapon, a way to poison enemies. It is also much simpler than the flagellum, built from about 10 proteins. Several of those proteins are near identical to the proteins at the base of the motor. This is what biologists call homology. The genes that build the needle are cousins of the genes that build the flagellum. Lay the DNA letters side by side and you see the family resemblance. So which came first? Did the flagellum grow out of the needle, adding new parts one at a time, each part useful on its own? This is the idea of co-option. Think of a hammer in a garage. It drives nails, but it also cracks ice, pulls staples, and props open a door. Same tool, new job. In biology, co-option means a part that evolved for one purpose gets borrowed for another. The needle's proteins may have worked in an ancient pump, a tube, or an anchor long before any propeller existed. Duplication, mutation, and reshuffling slowly turned old parts into new machinery. In 2015, a study in the journal Cell put this idea to the test. Researchers compared the genes of the flagellum and the needle across many species of bacteria. The result was not clean. The two systems did not descend in a straight line, one turning into the other. Their family tree split far earlier than expected, and each branch developed its own innovations. The needle did not simply become the flagellum. But the study did not kill co-option. It only showed that the history is messier. The proteins remain clearly related. They still share a deep common ancestor. Old parts were still recycled, just over a longer timeline with more detours. So where does this leave irreducible complexity? The motor still spins, but its parts were not always motor parts. Some were needles, some were pumps, some were structures that no longer exist. Each earned its place before the motor existed, and yet the needle is not a motor. The intermediate steps between weapon and propeller have not been found. The bridge is still missing, so the debate is not settled. The evolutionist showed that parts can change jobs. The design theorist showed that the final assembly still looks assembled. Between them sits a question no one has answered. How many steps does it take to make a miracle? But before the steps, there are instructions. The flagellum is built from instructions, and an instruction is a strange thing. In a dark room at Bletchley Park in 1941, rotors clicked as the Enigma machine scrambled messages into random-looking letters. The men listening made a leap of faith. If the noise is a code, there is a coder. They broke the code and found the coder. That single assumption would later ignite the greatest philosophical conflict in biology. Perry Marshall is an electrical engineer turned marketing consultant, not a biologist. When he first saw the table of the genetic code, he thought he was looking at ASCII, the code computers use to turn numbers into letters. In ASCII, the number 65 means uppercase. Nothing about ink or paper makes that necessary. It is an agreement, like friends deciding that the word green shall mean happiness. In DNA, the triplet TTT means phenylalanine. GGG means glycine. No chemical law forces those meanings. Marshall said, "A code with arbitrary meanings requires a chooser." Joshua Swamidass, a computational biologist at Washington University, pushed back. The genetic code is not fully arbitrary. Amino acids with similar chemical shapes share similar codons. A single-letter mutation often swaps in a similar amino acid, softening the damage. Chemistry has shaped the code. If you want to argue for a designer, you cannot lean on pure arbitrariness. But here is the deeper cut. Even a constrained code is still a code. A CD is plastic with pits. The music is not the pits. A book is paper and ink. The story is not the paper and ink. DNA is a molecule. The information is not the molecule. Chemistry might explain the pattern of the mapping, but it does not explain how a physical thing carries a non-physical thing. It does not explain how symbols come to mean anything, so the room goes quiet again. If the code was chosen, we find a mind. If it was not chosen, then matter somehow gave birth to meaning, and we still do not know how. And if the code is both chemical and symbolic, we are standing at the exact point where physics becomes something more than itself, and that something remains unnamed. Everything we have seen so far, code meaning, the flagellum's assembly, begs a raw question. Before any instruction existed, where did the first instruction come from? Life has a chicken and egg problem. DNA carries the blueprint for proteins, but DNA cannot copy itself. Proteins build and copy DNA, but proteins are made only by reading DNA. Each needs the other. If both must exist for either to work, life seems impossible to start. But there is a third molecule. RNA looks like DNA but is not just a library. It can fold into shapes and perform chemical reactions, like a protein, and it can carry information, like DNA. So scientists asked, could RNA have been the first living molecule? One molecule that did both jobs, before DNA and proteins split the work. For decades this was just a guess. The problem was replication. RNA alone does not copy itself easily. Then a lab at Ludwig Maximilian University in Munich ran an experiment. They took transfer RNA molecules, tiny workers that normally carry amino acids, and placed them in a tube. Then they cycled the temperature. Heat pulled the strands apart. Cold let them rejoin. Under those repeated swings, the RNA molecules began assembling each other, forming copies from fragments. No proteins, no enzymes, just heat and cold and time. The molecules built a self-replicating system from the physical rhythm of the room. What does that prove? It proves that chemistry alone can produce a primitive form of heredity. The first step might not have needed a designer, but here is the unopened door. RNA can replicate, yet it is fragile and error-prone. DNA is stronger, but needs proteins. Proteins are versatile, but need DNA. At some point, the RNA world had to hand over power to the DNA-protein empire we live in. How did that happen? The ribosome, the machine that reads RNA and builds proteins, is partly made of RNA, a relic that suggests the handover happened. But no one has recreated the steps in a lab. No one has shown how a tube of self-copying RNA becomes a living cell with a genetic code. The transition remains invisible, so the mystery deepens. We have a molecule that can carry meaning and do work. We have a physical process that can copy it. But between them lies the same gap we keep circling, the moment molecules begin to mean something. And we still do not know where that meaning comes from. We have followed this thread from the enigma machine to the ribosome, and it has led us to a cliff. On one side stands Stephen Meyer, a former geophysicist who spent years staring at the genetic text and concluded that DNA looks exactly like something an engineer would design. He wrote a book called Signature in the Cell to argue that a code demands a coder. On the other side stand those who say chemistry, given billions of years, can produce anything that looks like a code. Both sides look at the same molecule, both see the same triplets, and they read them in opposite ways. That disagreement is not scientific, it is philosophical. Science measures molecules, but it cannot measure a purpose. You cannot put a designer in a test tube. The universe looks identical whether a mind wrote the code or chemistry fumbled into it. Every experiment, every observation, comes back the same. So the question swings open into a space where science has no authority. And that is where the emotional weight lands, because this is not an old puzzle about ancient chemistry, it is personal. If DNA is written by a mind, then you are written. Your existence is an intended message. If the code is an accident, then your feeling that you matter might be an accident too. One possibility offers you a parent, the other offers you an orphanage, and the evidence does not firmly hand you either. Here is the honest truth. No experiment has detected a designer. No formula proves that consciousness wrote the first code. We have a molecule that stores language, a machine that reads it, and a gap between them that no current tool can cross. So you are standing at the abyss, not the abyss of emptiness, the abyss of unanswered questions. Philosophers have stood here for thousands of years. Scientists have now joined them. And the only honest sentence available is this: something in this universe behaves like an author. Whether an author exists behind it, we cannot yet say. Thank you for walking to the edge with us. The question does not end here. It lives inside every cell of your body. Where do you stand? Mind, chemistry, or somewhere in between? Write your answer in the comments and let us keep thinking together at the edge of the unknown. We have reached the edge, and the edge has a view. What we know: DNA is a code that builds machines, and some of those machines cannot be simplified. What we do not know: where the code came from or whether a mind wrote it. No test tube has written meaning from nothing. No microscope has caught a designer in the act. So we return to the twisted ladder, the molecule that stores you. It waits. It does not explain itself. Is it a signature or a coincidence wearing a signature's clothes? The answer has not come. Maybe it never will. Thank you for climbing this far with us. If you carry your own answer, the comments below are open. The search continues.